Hi everyone, welcome to the lecture on sex determination and sex chromosomes. This is perhaps one of my favorite subjects to talk about, and I think we'll just jump right on in, but it's fascinating to me. It's an area of research that I spent some time during my master's degree, which is in pharmacy and pharmaceuticals, but specifically it focused on sort of the forensic aspects of DNA and serology and tests that we would use to determine not only sex and paternity, but all kinds of criminality, like who may have been at the crime scene and that sort of thing. So it really, my master's program really delved into the analytical testing and understanding of how our bodies, genetics and DNA are give us information about things. And so this was fascinating to me, and I did my final thesis on how, my basic thesis was there isn't any one test that can unequivocally determine sex. There's ambiguity that can at times not be resolved. So you might think in your head right now, well, if their chromosomes are X and Y, then clearly they are male. We will talk about that, and it's clearly not the case, as you will learn in this lecture. Now, that said, if you are a boy or you are a girl, I am not challenging that. You are probably whatever you think your sex is. 99% of the time that's true. That's not a hard statistic. I made that up. I'm not sure what the certainty of sex is. However, I can say that in situations of ambiguity, there isn't any one way that can say definitively whether somebody is a male or female. So hang on to that thought as we work through this. So a lot of what we're going to talk about is the genetics of how sex is determined. And in general, sexual variety and what we call dimorphism are very important in the maintenance of species and our evolutionary histories, both our species and other species that we share the earth with. Sexual differentiation or dimorphism is a term that is used to describe differences between the sexes. And sometimes it's very obvious, and sometimes it isn't. I do a lot of bird banding in my life, and some birds are very sexually dimorphic. An example here are these wood ducks. The female's kind of drab and the male is stunning, right? He's spectacular. And that's dimorphism, where the male and the females are not looking the same phenotypically. I'm going to try to use some terms of genetics. I ask you this question and you can answer it later. Do you think humans are sexually dimorphic? Are there clear phenotypic differences, despite people that have short hair and things like that? But, I think you know the answer to that. Again, some of these are social constructs. The long hair and those sorts of things are not necessarily the sexual dimorphic. We are both, sexes are born with hair, but what are sexual differences? You can think of some upstairs and downstairs that would describe this condition, facial hair and that sort of thing. Anyway, think about that as we proceed through this as well. But you can see by these wood ducks that the male is stunning, like I said, and this is an evolutionary advantage for that male, especially within its own species. The sexier he is to the female, the more color, the more she's going to think, hey, he might have good genes that I might want to get in on and pass on to my offspring. So it's the females typically in certain species, in particular birds in this case, that pick the male based on their prowess and their, could be color. Red-winged blackbirds have a little red patch on their shoulder. The brighter it is, the more successful they are at attracting a mate. So that's one way the males compete for mates, is try to be stunning. All right, so also many creatures in nature exhibit different chromosomes that are associated with sex differences within a species. We're going to kind of talk about that. Mostly we'll talk about human genetics, but I'll throw in a little bit with some other species as well. So anyway, one of the important concepts throughout all of our discussion with genetics should be that genetic variation is good. We like it. There are a few exceptions, but what genetic variety does is it contributes to the success of the species. It basically allows you to mix up your genes, good and bad, and hopefully in a situation that you would be, especially with antibiotic resistance, we can see that if there's a situation that challenges you, if somebody in that species can survive it by having a mutation that allows them to survive it, the species as a whole will survive. So genetic variation is really good for a species to keep it not extinct and not endangered and healthy of surviving to the in subsequent generations. And so a limited gene pool, if this is what's called the founder's effect. You notice it in communities like Amish and that sort of thing, where the group is kind of closed. They don't intermarry a lot, and so their gene pool is very limited. With limited gene pool, there's just the probability that a bad gene will show up is a lot higher. There are a few exceptions, and I've just learned this recently, where there is a mutation that does actually make things better in a closed community. So there's that possibility as well. But usually we start to think of lethal genes. And the bigger the N, the larger the population, i.e. the further away you are from a founder's effect, the better, the healthier the gene pool is. We've probably weeded out, or you're not going to come in contact with 2 recessive genes, like we talked about before, that might cause a disease. All right, so going on, let's talk about how sex is determined in humans. I think you all know how sex happens, but how is not gender, but sex assigned at birth? And you probably have all heard this in 6th grade science, that females have what are called 2 X chromosomes. We talked about this before. Autosomal chromosomes are those that are related to body and are not going to have much to do with sex, let's put it that way. The X and the Y's chromosomes are what determine sex, and those are the last ones that show up on a karyotype if you map it out the way you're supposed to. So the Y and the X are determinants of sex, and in simple terms, if you have 2 X's, you're going to be a female, and if you have an X and a Y, you're going to be a male. So you get this X in a male pretty much, came from his father, his mother. The Y choice here, he was going to get either an X or a Y, and he got a Y. So if you do a Punnett square with the X and Y across the top, signifying the genotype of a male, just at one set of chromosomes, which are the sex chromosomes, and you go the females down the side, which is XX, and you do your crosses. You should be good at these. You have an X and an X here, X and a Y here, X and X here, X and Y here. It's 50-50 chance, and it's always going to be determined. Again, this is the male, so he has an X and a Y. He is always going to determine the sex. So women out there, if you're married and have kids and you're mad because you have all 3 girls, you can blame your husband. Just making fun here and poking some fun at this all. But what's really going on here is, depending on whether or not during meiosis, every cell has an X and a Y, but during meiosis, it's going to get down to a haploid number, and one of those chromosome haploids is going to be the sex chromosomes, and it's going to have either an X or a Y in it during meiosis, those gametes. And so if then during fertilization, the mom is always going to give an X. You can see there's no other choice. It's a different X, and there's different genes and different heredity on that X. But if the Y is given during that successfully fertilizes the egg, you're going to have a male. All right, and you can notice, we're going to talk about this here, I think, in the next slide. I just kind of went through that. It seems like that there would be, we're going to talk more about this in the future, but it seems like based on this probability, it's 50-50. And I would say that's pretty close. That ratio is not exact. There's been some discrepancies in the numbers that we can actually observe in human populations, and it has led to a lot of research. And the short version of the story is it's pretty close to 50-50, with more males making it to birth. And it's probably, at one point, early on in my life, it was thought of that the Y containing sperm might swim a little faster and get to the eggs first, which might account for that slight variability that's observed in the human population. But that's not necessarily been proven, but it's a possibility. Other thoughts are that just during embryo formation, perhaps there's something, some sort of reason why the 2 X's are less viable. So we'll talk more about that coming up here. Other organisms in our world and other sex types of determination, there's some variety in that. There's a lot of organisms that are similar to us, where there's an X and a Y type chromosome or sex chromosomes. Sorry, I have a typo here. In other organisms, sometimes it's the female that actually determines the sex and not the male. And these are actual models of what are sex chromosomes for the human. You can see the Y is itty bitty, and it's really missing a lot of information, and that's going to come into play when we talk about sex-linked disorders such as colorblindness and hemophilia and male pattern baldness. Those basically, these are what are called homologous chromosomes, except there's some meaning that they line up in the same place on in the cells, and they sort out function of genes together, like the gene for color blindness is found at the same location on one chromosome as the others. But in this case, let's say it's right here, hypothetically. Well, there's nothing here to offset that. That's why in cases of sex-linked recessive disorders, those that are related to the sex chromosomes, there isn't an offsetting dominant possibility out there. There's just nothing on that chromosome. And so it would only take one recessive in sex-linked recessive disorders. All right, so other versions of sex, there's some butterflies that have 2 X chromosomes and 14, the females have 2 X's and 14 totals, and the males are just missing, they only have 1 X and 13 total chromosomes, so they don't have a balanced number. And so it's determined by 1 less X chromosome, basically. And it's kind of efficient in this way, in a weird way, because they don't have to, quote, inactivate an X chromosome like we do, which we talked about. Basically, 2 X's are kind of a little bit, it's a potent dose of X, and we'll talk about that in a minute. All right, so let's look at some chromosome mutations that we talked about before. We talked a little bit about meiosis and how you can have extra chromosomes in one of your, we looked at trisomy, where you have 3 chromosomes due to non-disjunction instead of 2 in a cell in a fertilized embryo. So what happens in non-disjunction is during meiosis, the goal is to get to each gamete having 1/2 the number of chromosomes. Well, sometimes during that separation to form that, instead of one going this way and one going this way, both end up going together and 0 are over here on the other side. So instead of, when you're making your egg and sperm, instead of equal numbers getting distributed, one sperm is going to get 2 copies of a chromosome and the other sperm is going to get 0. And if that sperm, and again, there's a bazillion sperms, so this is all random chances, but if a sperm with, it's not void of all chromosomes, it's just missing 1, chromosome that went this way. So the rest of the chromosomes are there, 1 is missing. If it fertilized with a female with 1 in it, together they're just going to have 1 and not the complement, what we would expect to have 2. That's called monosomy. If the sperm with 2 of the extra chromosome fertilized an egg with 1, you're going to get 3. We call that trisomy, and that's Down syndrome. And these are all results of errors of what we call non-disjunction, where it didn't distribute evenly, basically. So anyway, yeah, I just talked about that. So if this happens with a sex chromosome, it becomes interesting. Mostly, we have 22 that are not sex chromosomes and 1 pair that are sex chromosomes. But if it does happen with sex chromosome, and you have a person that only has 1 X, the egg or sperm made just, there was a 0 in the sperm or egg, and then there was just 1 X that was used, whether it came from the male or the female, it doesn't really matter in this case. But you get a total of 45. That's how you would write it, 45X. And there's not 2 X's. It's just 1 that's missing 1 of those chromosomes. And normally there's developmental differences of stature and fertility and those sorts of things, but they present as a female. There's a classic, well, I don't know about classic, but I think she was on NCIS. I forget her name. Hunt is her last name, and I'm drawing a blank on the first. But anyway, she's an actress that has Turner syndrome. So you can live, but likely not reproducibly, like you cannot reproduce easily. So let's look at some other situations of chromosome number abnormalities. Anyway, here's another example of a young lady with Turner syndrome. There's just short stature, low hairline, different constrictions of the aorta, poor breast development, elbow deformity, no menstruation, brown spots, weird stuff, and it's honestly hard to explain why some of these things happen, but this girl is, she's pretty normal too. It's not that it's horribly obvious and dysfunctionally, it doesn't limit what a person can do necessarily. And it just really will all depend. And anyway, there's another situation with females called triple X, and I will caution you to not look that up. and in the form of XXX, or you'll get pornography on the computer. But if you look up the word triple X, we would write it this way to show the genotype. So instead of 46 total chromosomes, we have 47, and where we have the extra one is the X. This is similar to Down syndrome. It's trisomy. It's just with the sex chromosome. It happens about 1 in every 1,000 girls. The phenotype usually presents normally female. There's some delayed secondary sex characteristics, and sterility is possible, and maybe ADHD, and sometimes larger bodies. And anyway, we're going to talk about this in a bit in this slide, but they do have X inactivation, which I haven't talked about yet. So two of those X's are kind of going to get inactivated, but they're still, so developmentally, you probably wouldn't notice. I actually have a friend whose niece has trisomy of the sex chromosomes. She's triple X, and you would never know unless you were tested. And some people probably have this and don't know, because why would you be tested? Why would you look for your genetic makeup? But some people, there's problems during pregnancy or amniocentesis and those sorts of things, you could find out about it. All right, so what about males? Or this is ambiguous, right? We sort of associate this with male because they sort of present phenotypically as male. It's called Klinefelter syndrome. They have 47 chromosomes, similar to XXX, triple X, but they have XX and Y. So they have a dose of female and a dose of male. And so it's trisomy again, because there's 3 instead of 2. They present phenotypically as male, but this extra chromosome, one of them will get inactivated, leading, we haven't talked about X inactivation yet, but it's a normal part of every girl too, where one of the chromosomes sort of gets turned off in order to control dose and to compensate for dose, because 2 X's is very potent, so to speak. So anyway, the extra X doesn't activate, and because of that, there's a variety, depending on which X inactivates, there could be a variety of different symptoms for these individuals, but generally they're taller with longer arms and legs and hands, and a pretty normal, again, we might not know about them and it's ambiguous. This is exactly what I was talking about. If you had somebody with 2 X's and a Y, or 3 X's and 1 X, and in this case, X and 2 Y's, it's a very interesting, I don't think it has a name. I remember hearing about it a long time ago myself, but if there's an individual with 47 chromosomes total, and the trisomy occurs in the sex chromosomes where you have an extra Y, it's going to be kind of associated with aggressive males, because the Y doesn't really inactivate. And so it was first observed in 1965 in prison populations, and it was really noticeable in those that have had violent offenses. And so I'm not sure what the frequency is. I could have found that. I just forgot to look. But again, you wouldn't necessarily know unless you went looking for it. So it's very interesting stuff. All right, let's look a little bit closer about the X and the Y's, and we've talked a little bit about this already, and I will talk more about X inactivation, but you can notice this is from a karyotype, and you can see a female who got 1 X from her mom and 1 X from her dad. Those are homologous chromosomes. These are homologous too, except the one that areas of it are not, because they're not there, and that's the short version of the Y. This was given by the dad. So again, the male is going to determine the sex, whether or not he gives an X or a Y. This missingness, so to speak, causes a lot of, not a lot, there's just missing genes. And so, like I was talking about with hemophilia and male pattern baldness, this is classically a sex-linked sex-linked recessive disorder, where it would only take 1 gene, because there's nothing over here to play the role of the dominant gene. So if a typical, if a gene that causes a disorder is located on the sex chromosomes and part of it's missing, it's just going to take 1 recessive to phenotypically manifest that condition. And male pattern baldness, hemophilia, and colored blindness are examples of that. So we'll leave that. You should also notice that having 2 X's, as I've been talking about this thing called X inactivation, it's quite a heavy dose of female genes. Because of that, as such, randomly and variably, 1 is inactivated sometime after fertilization. It's not immediate. So you get the egg, the 2 X's join, and there's an embryo that starts to form by cell division. At this point, now we're doing mitosis, and Eventually the cells will differentiate. At some point, X's in cells will inactivate, and then every cell from there will carry that inactivation and expression forward. And so you can, it's really interesting. And essentially, I mean, I find X inactivation fascinating. In essence, females are a mosaic, and you can really conceptualize this if you think of female cats, and they are only, females are typically calico cats, and it's because of X inactivation. So you'll see a patch of orange fur because the gene that codes for orange color is on one X, but in another spot, the X inactivation occurred for the orange, and so now you get a splash of black or another color. So it's really fascinating, and females are actually mosaics. And anyway, there's some advantage to that, which is kind of conflicting to the ratio issue that I'll talk about later. But there are 1,100 genes on the X chromosome, and most of them are not expressed from the Y chromosome. And so I'm a little redundant there, and I apologize. So this kind of makes men a little bit more vulnerable than females, because any mutation that affects the gene on their X chromosome would affect basically their only copy of that gene, because there's nothing on the other side of that. So for me, I have a mutation from my dad, right? I have BRCA2 mutation for breast cancer. I got that from my dad, but I got a good one from my mom, and that's good. And so during most cell divisions, my dad's never works, but my mom's takes care of the business, and it works. If at one time my mom's doesn't work, then I don't have a backup. That's basically what this is saying, because there's no area on that Y. It's part of, and if the X inactivates, sorry, I got that. I was talking about another effect of the not having the recessive part, but back to my point about X inactivation. So if the X that had the good gene, like from my mom, is inactivated, then I'm stuck with my dad's gene. So it's not the case with breast cancer, because the BRCA2 is not carried on this sex chromosome, but that's the kind of the point that I'm trying to make. Hopefully that was understood. It was a little sloppy, but I think you got it. So in summary, X inactivation is to try to control dosage and to compensate for it, so that we don't have a double dose of genes in a highly penetrable dose of dangerous or expressive genes. And so when an X chromosome, is inactivated, it is called a bar body. And these are some pictures trying to depict that. They're in your book. All right. As I mentioned, the calico cats are pretty cool, and it's just simply what I said. The genetics of cats are simply fascinating, especially when you get into, Siamese cats and those sorts of things. Temperature kind of affects their expression of the color and their points, and because it's cooler temperatures up here, and we have some other temperature effects that I'll talk about later. But in short, it's all about X inactivation. If the X with the orange allele is active, that patch of skin is going to show orange. Vice versa. If the orange allele is compacted into the bar body, the X black allele is active and will show. And so that's why a cat is calico cat's a female. Very, very cool stuff. And this is a good, so 3 colors is basically what you would get for a calico cat for it to be female. Very cool. I have a cat that looks a lot like that. All right, so X and Y's in development. This is really cool. Very cool stuff here. So we all began sort of as females, or if you want to say hermaphrodites, it's ambiguous. So we have our chromosomes, but how we develop has not yet been fully laid out. And so at about 7 weeks, and I might be off a little bit here, 5 to 7, somewhere in there, after fertilization, the Y gene kicks in if it's a boy. And it'll start to say, okay, you're developing as a female, but I see this X, and there's all these chemicals and hormones and signals coming from this Y, from the Y chromosome, and now I want you to start making a boy baby. And I'm talking to myself as myself is creating myself, cells. And so basically the Y signal, if it's present, will start to signal for the development of the boy. And so this female will then sort of respond to this chemical signaling, and it's actually from a gene called the SRY gene on the Y chromosome, and they'll start to make a male. Here's the catch. Like I said before, I'm not sure if this is where we're at in our lecture, but there's this thing called androgen resistance, and or androgen sensitivity and insensitivity and those sorts of things. Basically, nothing in nature is perfect, right? And so some XY individuals actually develop into a female instead of a male because during development, that SRY gene and its influence is resistant and rendered rather useless. And these individuals develop into females, but they're genetically male. And this caused quite a controversy, and it still is undecided and unresolved in the Olympics. The question is, where would these individuals compete? So what you basically have is a genetic male, X and Y, but the Y basically useless, and so the person develops as a female. This is an example. This is a runway model. I forget her name, but anyway, she came out because there were many questions about her gender or sex, sorry, and she admitted to being XY, but was had androgen resistance, so she developed as a female. This is Caster Semenya, probably one of the more famous cases because she's challenged the Olympics. This is a developed woman who has an XY. Well, I think we don't know for sure exactly, but there have been leaks in the press that she is genetically male, but she developed as a female and competed as a female and competed better than the other females. And so in high level stakes of Olympics and that sort of thing, it became a question. And they basically have been dealing with more than just Caster. It was back in the 70s too with some German, I think it was German, maybe Austrian, I'm not sure, but there were some other athletes that are female that were really XY males. And they have put, the Olympic Committee and others have put female athletes through ridiculous scrutiny, asking to show genitals and submit to testing to prove their sex. And like I said earlier, that's not a slam dunk. And so anyway, like, how do you make it fair then that girls are competing against girls? What defines a girl sexually? It's a fascinating reading, and it's currently going through the Olympics as to like, how do we do this? At one point, they decided to try to use testosterone levels and then determine, well, that's not fair because testosterone is a controlled substance. And what if you just naturally have more because of something like this? And so even though there's no why, there is some not, so genes are not, it's not a perfect, maybe not everything is resistant. So there's some parts of the Y that might come out because genes sort of, even during X inactivation, maybe not every gene is inactivated. So we don't know everything. But anyway, in this case, it was proved to be very interesting going through the committee and competition where it really matters. So what's so special about the Y? It was thought at one point not to have any, if any, genes, and then the Human Genome Project helped identify 75 genes on the Y chromosome. Compared to the X, it has a lot more, 900 to 1400. And like I said, some of these are homologous, some are not, meaning you have a complementary kind of counterpart to it, but obviously by sheer volume you can't, because there's not enough to go around. And there's this area called the pars, or the pseudo-autosomal area, where there is that, where the homology would exist, where there's some matching. We've already talked about this, that sex-determining gene is SRY. It's the one that expresses it, actually, phenotypically. The Y kind of determines it, but the gene itself has to sort of come into play, and that's where we would get some of the androgen resistance. Some males with two X's and an SRY area on their X, some males actually have two X chromosomes, and they develop as male because of this SRY area on the X chromosome got recombined with the X chromosome. And so that's kind of another interesting ambiguity that I didn't actually talk much about in my master's thesis, but it's another interesting one. Sometimes you just can't pick up the Y gene in testing. But anyway, some females actually have an XY, and where the Y is missing the SRY gene, which would lead to the androgen sensitivity or resistance. So a little bit more left on this lecture, but again, it's one of my favorites. So males and females, what's the dealio there with the ratios? Like I said, the ratios are not exactly 1-to-1, and they don't know everything about this. Quite frankly, the X and the Y, when I just talked about that other situation where you would have two X's and a male might develop because of recombination, so the parts of the Y actually ended up on an X because there is some recombinant shuffling of the genes during mitosis and meiosis. So they do snap at certain areas and then segregate into different gametes. And so there could be some possibilities for the ratios to be off because of that. You would, again, you would expect a 1-to-1 based on that Punnett square and just what we see in life, but it's a little different, and why is actually not well known. There's some theories, and really in my world, it doesn't matter too much. There seems to be an equal number conceived, but more males kind of make it to term. And they did some studies, I think, with some miscarried material or, embryonic material. I can't remember exactly what it was, but they did sort of decide that it seems like the numbers of conception are equal, but more males make it to term, and they don't really know why. So there's different ratios that they talk about, secondary and primary sex ratio. But the primary sex ratio used to favor more males, but this dogma has sort of been replaced. And they used to think it's because more males were conceived, but now they think it's about the bringing to term. And the theory now that's sort of central dogma to this is that female mortality before birth, so less female embryos actually make it to term, is sort of the current thought. They used to think it was because the conception numbers were different, maybe the swimming wise, but now it seems to be this is where research is leaning, but there's more research that could be done on this. So back to the line of the X chromosome inactivation, they were trying to figure out, researchers, how do we know which X, what accounts for which X is inactivated or not? And they basically, the line hypothesis basically says that it's random and at any point early in embryonic development. And then obviously all the cell progeny would have inactivated X, the inactivated X bar body and the activated X, and are sort of a mosaic. So basically all of the line hypothesis proved is that there is no rhyme or reason. It's completely random and somewhere early in development. Really fascinating stuff. All right, the mechanisms of an X inactivation. One of the things that we talk about is imprinting, which is not what you're thinking about, how a baby chick will learn to imprint to me and think I'm its mommy, but more imprinting of DNA memory. And how does this commitment of X inactivation happen in cellular memory? And they did discover that not all of the entire X is inactivated, sometimes just the genes. And a lot of this has to do with some transcription factors in this fascinating field that we will talk about called epigenetics. We'll get to that later. But basically, there's part of your DNA that codes for proteins and kind of expresses you. And then there's part of your DNA that regulates that process. And that's what epigenetics deal with. It's the turning on and off of genes. And that's really where the environment influences your genetic Mendelian makeup. So very, very cool stuff to come. One of the questions I'm probably going to ask you is on page 92 of the book. If you don't have this book for some reason, look up carbon copy and cats and genetics. And basically this little fella girl, I can't remember, I think it's a girl, because this cat is a clone of this cat. How are these different? I mean, why wouldn't you expect the same coloring here? So if you can explain carbon copy, you have a pretty good understanding of this lecture. So I'll leave you to ponder that. One more couple things about other organisms other than humans, Drosophila and C. elegans. C. elegans is a nematode, it's a worm, and it's kind of genetically dumb. It doesn't have many cells, and it doesn't have a Y, but the Drosophila, your basic fruit fly, has a Y, but that Y has no role. And so how is sex determined in organisms like this? And we've already talked a little bit about the butterfly, but what's this part of the book and the lectures trying to explain is how sometimes the ratio of the X chromosome to the actual number of autosomal chromosomes is a factor. And if you look at this, it's all, well, before I get to what I was going to say, the Drosophila have a Y, but it doesn't seem to play a role. Instead, the sex determination of a Drosophila is really variable within ranges, and it's based on the influence of other autosomal chromosomes that help determine sex. And they've done a lot of cross studies. So think of Mendel and his peas. Now Drosophila is another organism that turns around quickly from generations. And so they did a lot of cross studies to come to these understandings, and they learned that there's a lot of variation in Drosophila sex from super female to super male to intermediates. And so this all had to do with the genetic balance theory. But if you look at the chart, I'm gonna move myself down here for a minute. This is kind of hard to understand, but the basic understanding is the ratio of X chromosomes to autosomal sets. The higher the number, the more female. The lower the number, the more male. So basically, the X's and the Y's are influenced by the autosomal chromosomes, is what this is trying to show. So back to continuing on with the nematode worm. It's a very well-studied worm because of this simplicity in a way. It's got a very precise lineage because it doesn't have many cells. And one of the things that's really interesting in when you study genetics is controlling the cell lines. And so this has very specific embryonic origin. So we know a lot about it from a evolutionary perspective, and there's not a lot of garbage to sort through. So it basically has two sexual phenotypes. Males get one X and hermaphrodites get two. And then when needed, the egg oogenesis sort of occurs in these hermaphrodites as the adult sort of matures, and then there's fertilization. But so just a couple of other examples from nature. All right, the last thing we'll talk about is temperature, and I put this on there seriously. Yes, temperature-dependent sex determination is a thing. It is found in a lot of reptilian species where sex determination can sometimes be determined by chromosomal genotype, but other times it's temperature. And it probably is related to an enzyme, but there's a lot of research being done all the time on this. But crocodiles, your basic crocodiles, turtles, and some lizards, again, they find it related to egg incubation, and it's protective, I think, in some way. So if there's a low temperature around, it's there's certain species that will develop into be 100% females, and high temperatures 100% males, and intermediate temperatures are variable. But what is really interesting is why, and I think it's the activation of necessary enzymes that are involved in sex determination. Your enzymes in your body, like a certain body temp, like your body temperature is tightly controlled, and the reason that is because your biochemical processing, your biochemistry to do chemical reactions, the temperature's got to be right. And we all know catalysts sort of change the reaction times and conditions to speed up or slow down a reaction, typically speeding it up. And enzymes are catalysts; they're biological catalysts. And so there's an enzyme called aromatase that converts testosterone to estrogen, and it's affected by different temperatures. And so a lot of the thought is around that so what if you're not converting testosterone to estrogen, you're going to have more testosterone and develop male. This enzyme is actually, interestingly so, the same enzyme that is often blocked in some breast cancer treatments, because breast cancer and other cancers that are driven by hormonal growth factors like testosterone and estrogen, sometimes you just need to block it. Like prostate cancer, testosterone will feed that cancer because testosterone is a growth factor, right? You're going to big, build, and strong. Same with estrogen. So you try to block the action of estrogen. Well, in women that have exceeded menopause but still have cancer or still need to block the estrogen that is made elsewhere, this is blocked. This enzyme is blocked actually so that we don't convert some of our testosterone into estrogen. Anyway, it's a complicated thing, but it's just an interest part of this story. So I believe that's it. All right. Thank you, and see me if you have questions.